In this session, we are going to learn about the anatomy of dicot stem. So let us begin. So let us understand the anatomy of dicot stem with the help of a diagram. So first of all, there will be a layer of epidermis. Now, since this is a stem, the epidermis will have cuticle over it, a layer of cuticle to prevent water loss. We already learned about the cuticle, which is a waxy coating, and it is present over the epidermis. Usually, cuticle is found in the epidermis of the stem so that the water loss can be prevented. Now, the epidermis may also have a few stomata. Now, below the epidermis, we will get a hypodermis. Hypodermis is present below the epidermis, and this hypodermis is made up of collenchyma cells. Alright, so this collenchyma provides mechanical strength to the young stem. Now, below the collenchymatous hypodermis, there will be several layers of round and thin walled parenchymatous cells with intercellular spaces between them. This constitutes the cortex. So the cortex is parenchymatous, then below the cortex we will get the last or the innermost layer, which is the bundle sheath. I'm sorry, not bundle sheath, the endodermis, the innermost layer of the cortex. So we will have endodermis here, and the endodermis has, the cells of the endodermis has starch grains. These starch grains are present in the cells of endodermis, and therefore this layer of endodermis bearing starch cells is called starch sheath. If you remember, when we learnt about the anatomy of a dicot root, there the endodermis had Casparian strip. Here in the case of dicot stem, we will find starch grains in the cells of the endodermis, and this layer is also called as starch sheath. Now, below the endodermis, there will be pericycle, but pericycle will be in the form of semilunar patches like this. So there will be semilunar patches of pericycle. Let me draw it properly. These semilunar patches of pericycle is made up of sclerenchyma. If you remember the anatomy of dicot root, the pericycle was continuous, but here in case of dicot stem, the pericycle is in the form of semilunar patches and is made up of sclerenchyma. This semilunar patches of pericycle is present just above the xylem. Now the central location is the pith. This is the pith, which is made up of again parenchyma cells with intercellular spaces, and in this pith there will be radial arrangement of vascular bundles. These vascular bundles will be arranged over the pericycle like this in the form of a ring. So this ring arrangement of vascular bundles is a characteristic feature of dicot stem. Alright, so these vascular bundles will be arranged in the form of ring, and these vascular bundles are since this is a dicot stem, so the vascular bundles are conjoined. That means xylem and phloem are present together in a bundle. Open, that means cambium will be present between the xylem and phloem, and the protoxylem is endark. That means the protoxylem will be towards the pith, right? So the vascular bundle is conjoined, open with endark protoxylem, and the vascular bundles are arranged in the form of ring. This ring arrangement of vascular bundle is the characteristic of dicot stem. Now between these vascular bundles there are radially arranged parenchyma cells in this portion. So this portion is called medullary rays. The central portion is the pith with parenchyma cells having intercellular spaces, and the pith is large and well developed. Alright, so this covers the anatomy of dicot stem. Now let us look at the anatomy of a monocot stem. Alright, I hope this one is clear. So let us move on to the anatomy of monocot stem. So first of all there will be epidermis as usual. Epidermis may have cuticle and trichomes. Did I mention trichomes here? Okay, I forgot. Sorry, I forgot to mention that the epidermis also has trichomes. If you remember, we studied about trichomes. Trichomes are the hair which are present on the epidermis. Usually the hair of the epidermis in cases of stem are called trichomes, right? So this is a trichome. It is a multicellular. Trichome will be multicellular, right? In cases of roots we find unicellular root hairs. In cases of stem we find multicellular root hairs which are called trichome. So I'm not drawing trichome and cuticle here, just the epidermis. So this is the epidermis of the stem. Then we have a sclerenchymatous hypodermis. The hypodermis here is made up of sclerenchymatous cells, whereas in case of dicot stem it was made of collenchyma, right? So this is hypodermis made up of sclerenchyma. Now the rest of the part is very different from the dicot stem. If we look into the anatomy of monocot stem, it is quite unique, and you can easily identify that this anatomy represents a monocot stem. How? So the whole of this ground tissue, consists of parenchyma. Alright, so this parenchymatous ground tissue covers the whole internal structure of the monocot stem below the hypodermis. So the ground tissue is parenchymatous, and in this ground tissue we will find scattered vascular bundles. So the vascular bundles will be scattered in this ground tissue in a random fashion like this. The vascular bundles which are present at the center of the stem are usually larger as compared to those which are present towards the periphery like this. So the vascular bundles at the sides will be smaller and at the center will be larger, and the vascular bundles are scattered in the ground tissue in this manner, and each of these vascular bundles will be surrounded by a bundle sheath which is also made up of sclerenchymatous cells. So all these are unique features of monocot stem. If you ever look into the cross section of a monocot stem, you can easily identify it because of its unique characteristic features, right? So all these vascular bundles will have a covering of sclerenchymatous bundle sheath. This bundle sheath is made up of sclerenchyma. So both the hypodermis and the bundle sheath has sclerenchymatous cells here. Alright, and the vascular bundles, if we talk about the type of vascular bundles here, the vascular bundles will be closed. Since it is a monocot stem, so there is no possibility of secondary growth, right? So there will be no cambium, hence the vascular bundles are conjoined and closed. So conjoined, closed, and the type of protoxylem would be endark only, because in cases of stem we find endark protoxylem. Inside these vascular bundles we will find water containing cavities like this. Alright, so all these characteristics are quite different from what we saw in the dicot stem. Water containing cavities will be present inside the vascular bundles. Water containing cavities. And phloem parenchyma will be absent in cases of monocot stem. Alright? Phloem parenchyma is absent. Phloem parenchyma is a type of parenchyma tissue that we studied. It will be absent in case of monocot stem. So this completes the anatomy of monocot stem. It is quite unique, so I think it is easier for you to remember it. In case of monocot stem, we will have the hypodermis, which is made up of sclerenchyma. Then whole of the ground tissue is It is not divided into the cortex, endodermis, pericycle. None of it will be found here. Only the parenchyma is ground tissue covering the whole internal part of the stem, and in that ground tissue we will find the vascular bundles scattered. These vascular bundles will have water containing cavities, and they are conjoined and closed type. These vascular bundles will be covered by a bundle sheath made up of sclerenchyma. Alright, so this was about the monocot stem. Phloem parenchyma will be absent, and we already studied about the anatomy of dicot stem. In case of dicot stem, the characteristic feature is the radial arrangement of vascular bundles, and the vascular bundles are conjoined open, because in case of dicot stem secondary growth will happen. So how that will happen? Because of the presence of cambium between the xylem and phloem inside the vascular bundle. That's why open type, and the protoxylem is endark. Alright, and the hypodermis in case of dicot stem is made up of collenchyma, whereas the hypodermis of monocot stem was made of sclerenchyma. So you can easily understand and remember utilizing these differences between the two types of anatomies of dicot and monocot stem. Alright, so I hope the anatomy of dicot and monocot stem is clear to you. That's all for this lecture. Thank you so much.